So while glutamate is our primary excitatory neurotransmitter in the brain, it turns out that GABA is our primary inhibitory neurotransmitter in the brain, which means the majority of synapses where you actually, which result, where the release of neurotransmitter results in inhibition, which would mean, of course, hyperpolarization, right? Increasing the charge difference inside versus outside the postsynaptic cell to take that cell further from, the threshold potential of -55, where it might be, able to initiate an action potential. It's typically GABA, which is the neurotransmitter. And, GABA is pretty closely related to amino acids as well. It is some GABA is actually remarkably sort of synthesized from glutamate. So I think I always find this interesting, like a yin-yang relationship between our primary excitatory neurotransmitter glutamate and our primary inhibitory neurotransmitter GABA. There's an enzyme called glutamic acid decarboxylase, or GAD, which catalyzes the decarboxylation of glutamic acid to form GABA. GABA acts, we're going to see, at postsynaptic receptors. There will be metabotropic targets, as there are for glutamate, but there are also obviously going to be ionotropic targets too, right? Because we're going to rapidly, hyperpolarize, a neuron, and you're going to have, receptors that have channels, that are sort of built into their very structure that can, when GABA binds, set up a hyperpolarizing, kind of response. Glycine is another kind of related neurotransmitter that also is inhibitory in the brain, but it's not as well studied actually as GABA. GABA is really our primary, inhibitory neurotransmitter in the CNS.